Welcome to this video on the restriction endonucleases, the molecular scissors. These enzymes cleave the inner phosphodiester bonds in a double-stranded DNA in a sequence-selective manner. Werner Arbor, Dan Nathans, and Hamilton Smith received the Nobel Prize in 1978 for the discovery of these enzymes. These enzymes evolved as part of restriction modification system which protects bacteria while degrading the foreign DNA. In this video, I will explore the restriction modification system, types of restriction endonucleases, nomenclature, characteristics, restriction sites, cutting patterns and frequency, subtypes and significance. So what is restriction modification system? Scientists observed that bacteria infected by a virus became resistant to later infections. This resistance was due to restriction modification system consisting of methylase and restriction endonuclease. Methylase adds methyl group to the host DNA selectively, making it resistant to restriction endonucleases, whereas the viral DNA remains unmethylated and it is hydrolyzed by the restriction endonucleases. Therefore, the restriction enzymes evolved as part of restriction modification system, providing genomic immunity to bacteria. So what are restriction endonucleases? These enzymes hydrolyze inner phosphodiester bond in a double-stranded DNA in a sequence-selective manner, and the sequence they select, they bind, they recognize and cut is known as restriction site. They generate DNA fragments of defined size and length, and they are used in gene cloning. They do not generate free nucleotide. They do not digest single-stranded DNA or RNA. Discovery of these enzymes has revolutionized the field of molecular biology, particularly gene cloning and genetic engineering. These enzymes are classified into four types based on their subunits, cofactors, energy needs, and cleavage patterns. In this video, however, we will focus on type 2 enzymes, which are the most commonly used in genetic manipulations. These enzymes are more than 90% of the total restriction endonucleases known to date. Just to present an overview of type 2 restriction endonucleases, they are also known as type 2P because the restriction site of these enzymes is a palindromic sequence. These are the simplest, smallest, and the largest group of enzymes representing more than 90% of the total restriction endonucleases known to date. They are made up of one subunit having a DNA binding domain and a cleavage domain. They cut the target DNA within the restriction site, generating fragments of known length and size, and they are used in gene cloning or genetic manipulations. Subunit structure of restriction endonucleases. These enzymes have different subunit structures depending on the type of enzyme and the length of the recognition site they target. The enzymes that recognize 4 base pair restriction sites are often monomeric, having single subunit or polypeptide. Enzymes that recognize 6 base pair restriction sites are usually homodimeric, and the enzyme that recognize long site consisting of 8 base pair restriction sites are often homotetrameric, consisting of 4 identical polypeptide chain. Features of the restriction site. The site is a palindromic sequence which reads the same on both strands in the 5' to 3' direction. It may have 4, 6 or 8 base pair, however the hexameric site is the most common one. Did you ever think why is the restriction site palindromic? This may be for economical reasons because the palindromic site allows the same restriction enzyme to recognize and cleave both strands of DNA simultaneously. The cutting frequency and the total restriction sites, for example, in the human genome, can be calculated by knowing two pieces of information. The first one is you should know the size of restriction site. Second is you should know the human genome size. Human genome has 3 billion base pairs and the restriction sites could be 4, 6 or 8. So I have here two examples. First one is ALU1, which is a 4 cutter. The site is 4 base pair palindromic site. So it will cut every 256 base pair. The total sites in human genome are likely to be 12 million sites are likely to be present. The another example is a 6 cutter BAM H1 or AquaR1. Here this is BAM H1. This is palindromic site having 6 base pairs. It will cut every 4096 base pair, generating total 732,421 sites in human genome. So the frequency has been calculated using this formula. Frequency is equal to 1 over 4 raised to the power n. N is the size of the restriction site, which could be 4, 6 or 8, and total sites can be calculated by dividing the 3 billion base pairs by the frequency you calculated here. Restriction endonucleases produce two types of cutting patterns. First one is blunt end, second is cohesive end. So let me give you example of blunt end cutting pattern. So this is an example of SMA1. There are many other enzymes. This is a 4 cutter and the site where it will cut is shown by the arrowhead. So what is blunt end cutting? It is a cut straight through both strand of the DNA at the same position without producing any overhang. So this is called as blunt end cutting pattern. So let us discuss cohesive end cutting pattern. This is generated by enzymes that cut staggered, producing single-stranded 5' or 3' overhang. This is an example of AquaR1 which cuts this site at the point shown by arrowhead, and this produces 5' overhang. These overhangs are complementary to each other, therefore this is known as cohesive or sticky end cutting pattern or staggered end cutting pattern. Nomenclature. There are more than 3,000 different enzymes. They carry acronyms. I will explain the basis of nomenclature using two examples, AquaR1 and ND3. They are named after bacterial sources, genus, species, serotype, and the order in which they are discovered. For example, AquaR1, E here is genus Escherichia, CO is coli, species, R is the strain, and number one is the order in which it was discovered. Similarly, Hind 3H is Haemophilus is genus, IN is influenza species, D is strain, and number 3 is the order in which it was discovered. Based on the restriction site and cutting pattern, there are two subtypes of type 2 restriction endonucleases. First one is isoschizomers, second is neoschizomers. So let us discuss isoschizomers. Iso means same, schizomers means fragment or cutting pattern. There are 2 points which are similar, there are 2 points which are different. The first two different points are they are from different sources and they function in different conditions. Examples are SPH1 and BBU1. The similarity is they recognize the same restriction site which is given here, and the other similarity is they produce the same cutting pattern producing staggered end pattern. So let us discuss neoschizomers. Neo means different, schizomers means cutting pattern. I have selected two examples, APA1 and BSP1. The similarity is they recognize the same restriction site. Sequence is same here and here. There are three different points here about these enzymes. First one is the source is different. Second, they function in different reaction condition. Third one they produce different cutting pattern. APA1 produces 3' overhang, whereas BSP produces 5' overhang. They have enormous applications. They are used in gene cloning and gene mapping. They are also used in DNA fingerprinting or restriction fragment length polymorphism. They can also be used in disease diagnosis. Thank you for watching this video. I request you to like it, share it, and subscribe it. Until next time, you take care. Bye bye.